Hello students, in today's video we have to study about oxidative phosphorylation. Oxidative phosphorylation is a process by which the cell synthesizes its energy. Before watching this lecture, make sure that you must watch the previous videos from glycolysis to Krebs cycle, because oxidative phosphorylation is the final step of oxidative respiration, in which energy which is stored in the form of reduced form of coenzyme is released and it is converted into ATP, the energy currency of the cell. So let us look at the oxidative phosphorylation. If you look at the word oxidative phosphorylation, it is made up of two words, that is oxidative and phosphorylation. Oxidative means oxidation and phosphorylation means addition of phosphate group to ADP, that is adenosine diphosphate, to form ATP, that is adenosine triphosphate. In eukaryotes, oxidative phosphorylation occurs in mitochondria and it involves the reduction of oxygen to water with electron donated by NADH and FADH. NADH and FADH2 are the reduced form of coenzymes. When they get oxidized, they donate the pair of electrons that are transported by ETC to oxygen, and oxygen when the electron get reduced and combined with hydrogen to form water molecule, and this is coupled with the phosphorylation to yield the ATP. Before studying the ATP, oxidative phosphorylation, let us study about the structure where the oxidation phosphorylation takes place. As you know that oxidative phosphorylation takes place in mitochondria of the cell. Mitochondria, like gram-negative bacteria, is two-membrane system. It has outer membrane and inner membrane. If you look at the structure of mitochondria, you will find that it has outer membrane which is permeable to small molecules and ions. These molecules, small ions and molecules, get entry into the intermembrane space through the family of protein known as sporins. And second is inner membrane. This is inner membrane which is highly folded from cristae. On cristae we have ATP synthase and ATC complexes. This is the site of ATP synthesis, and inside the inner membrane we have matrix. Matrix is the site of Krebs cycle, and oxidation of various molecules like fatty acid oxidation and Krebs cycle take place in matrix of the mitochondria. Mitochondria also contain its own mitochondrial DNA. It has several genes which synthesize the protein required by the mitochondria. So mitochondria is a semi-autonomous body. Between the two membrane we have intermembrane space. It has its own function. We will discuss about it in upcoming slides. So how energy is produced in the ETC? Most of the free energy, as you know, that released during the oxidation of glucose to carbon dioxide is retained in reduced coenzyme, that is in the form of NADH and FADH2, generated during glycolysis and TCA cycle. Electrons are released from NADH and FADH2 are eventually transferred to oxygen, forming a water molecule. You know that these reactions, most of the reaction of oxidation, aerobic respiration of redox reaction, that is oxidation and reduction, take place. In this reaction, for example, NADH, when lose the electron, is oxidized to NAD, while as other half, that is oxygen, gain the electron released by the NADH, and it is converted into, when oxygen get reduced and it combined with the hydrogen ion and it form water. Likewise, the other reaction that takes place in this energy formation, that is FADH2, lose the electron and form FAD, and these electrons are gained by oxygen, and oxygen get reduced, which combine with the hydrogen molecule and form water. There are of there is two way of ATP formation. One is substrate level, and other is from this oxidation reduction of these coenzymes. And substrate level phosphorylation, the substrate transferred its phosphate to ADP and generate ATP directly, but this production of energy is very low. To obtain the large amount of energy, cell undergo the oxidative respiration, and generate these reduced form of coenzyme, and these coenzyme eventually release their electron via electron transport chain and produce energy, which we have to study in detail in upcoming slides. So let us study about the electron transport chain and its component. Electron pass through a series of membrane bound carriers. The mitochondrial respiratory chain consists of series of sequentially acting electron carriers, most of which are integral protein with prosthetic groups capable of accepting and donating either one or two electrons. For example, these are the components of electron transport chain. Let us take the example of NADH dehydrogenase. In this way, NADH, the question arises why electrons are transported from NADH to oxygen, because NADH has highest redox potential in negative, that is, it has 0.32 volt of electron potential, while as oxygen have highest positive electron potential, that is redox potential, that is positive 0.82 volt. So NADH has the highest tendency to lose the electron, while as oxygen has highest tendency to gain the electron. The electron from NADH, when NADH is oxidized, it releases the pair of electron. These electrons are transferred to the ubiquinone. Ubiquinone has the tendency to take two electrons. It can take two as well as one at a time. When it takes one electron, it gets reduced in semiquinone form to form semiquinone. When it takes the two electron, it can fully reduce to ubiquinol. Ubiquinol, when reduced, it transfers its electron to cytochrome B of complex 3, and ubiquinone, when transfer this electron, get oxidized and it can take further electron from NADH. When cytochrome have electron, it gets reduced and it transfers its electron to cytochrome C1. From cytochrome C1, electron are transported to cytochrome C. From cytochrome C, the electron are transported to cytochrome A, and to A they transfer to A3, and finally from A3 they are taken up by the oxygen, and this way oxygen get reduced. So let us study about the electron carriers. There are electron carriers function in multi-enzyme complexes. ETC has 4 complexes. Complex 1, also known as NADH ubiquinone oxidoreductase, or we call it as NADH dehydrogenase. Complex 1 is a multi-subunit enzyme. It is a large enzyme composed of 42 different polypeptide chain, including FMN, that is flavin mononucleotide, and at least 6 iron sulfur clusters. Complex 2, also known as succinate dehydrogenase. Succinate dehydrogenase is a electron move from in succinate dehydrogenase electron are moved from to FAD. FAD has to transfer their electron to complex 2. Electron move from succinate to FAD, then through 3 ferrous sulfur center to ubiquinone. Third complex is cytochrome BC1 complex or ubiquinone cytochrome C oxidoreductase. Cytochrome C is the acceptor of electron from quinone. However, quinone carries pair of electron. Cytochrome C can accept only one at a time. The last complex is complex 4, that is cytochrome oxidase. The fourth complex is composed of cytochrome protein C, cytochrome A and cytochrome A3 carriers electron from cytochrome C to molecular oxygen and reducing it to water. These are the complexes of electron transport chain. We will study about them in detail in upcoming slide. So until now, I hope you have familiar with the ETC components. So let us study the oxidative phosphorylation in detail. You know that oxidative phosphorylation takes place inside the mitochondria, and mitochondria is a double membrane system like gram bacteria. The outer membrane is freely permeable to the small molecules and ion through their channels of porine proteins. In between the inner membrane and outer membrane, we have intramembrane space. Inside the inner membrane, we have matrix where Krebs cycle and oxidation of fatty acids take place. We have complex 1, also known as NADH dehydrogenase. It contains one molecule of flavin mononucleotide and at least 6 iron sulfur clusters. Then we have complex 2. We call it as succinate coenzyme reductase or succinate dehydrogenase. It is very important to note that succinate dehydrogenase is the only enzyme which is bound to the inner membrane of mitochondria. It is the only membrane bound enzyme of Krebs cycle. Other enzymes of Krebs cycles are present in the matrix of the cell. Very important for those who are going to take their NEET examination or a NET examination. So it contains FAD. The electron transferred from FADH2 are accepted by FAD, and eventually these electrons are transferred to ferrous sulfur center clusters. And other molecules of ETC, very important molecule of ETC is a ubiquinone. Ubiquinone has 3 configuration, that is ubiquinone, semiquinone, and ubiquinol. When it accepts one electron, it converts into semiquinone, and when it gets the pair of electron, it converted into ubiquinol. After that, we have complex 3. Complex 3 has 2 important carrier proteins, that is cytochrome B and cytochrome C1. As we know that quinone has 2 electron, it can accept 2 electron, but cytochrome B can take only 1 electron at a time. Then we have another carrier protein, that is cytochrome C. Then we have complex 4, and complex 4 has 2 important cytochrome proteins, that is cytochrome A and cytochrome A3. These are the component of ETC chain. Electron from complex 1 and to complex 4 transferred via these carrier proteins. From the cytochrome A3, the electron are taken up by the oxygen, molecular oxygen. So next step, as we know that during Krebs cycle and oxidation of fatty acids, they produce NADH that is a reduced form of coenzyme. NADH has to be oxidized so as to produce NAD, so that these cycles and these oxidation reduction take place to produce energy. When NADH get reoxidized to NAD, it transfers a pair of electrons. The NAD is reused in the Krebs cycle or oxidation of fatty acids and other reactions. The pair of electron is accepted by a flavin mononucleotide, and from flavin mononucleotide it is transferred to ferrous sulfur cluster, and from ferrous sulfur cluster these molecules are transferred to ubiquinone. The transfer of pair of electron from complex 1 to ubiquinone, the complex 1 translocate 4 hydrogen ions proton to the intermembrane space. Likewise, in case of complex 2, succinate when converted into fumarase, the succinate dehydrogenase convert this reaction, and succinate release 2 electrons which are taken up by the FAD, and from FAD the electrons are taken up by the ferrous sulfur cluster, and from ferrous sulfur cluster the electrons are taken up by the ubiquinone. Ubiquinone in turn transfers this electron to cytochrome B. Ubiquinone can take 2 electrons, but cytochrome B only take 1 electron at a time. From cytochrome B, the electron transported to cytochrome C1. From cytochrome C1, the electrons are taken up by the cytochrome C. During this transfer from cytochrome B to cytochrome C, the complex 3 translocate 4 hydrogen molecules, hydrogen proton into the intermembrane space. When cytochrome C take the electron, it get reduced and it transfers its electron to cytochrome A of complex 4. The cytochrome A transfer these electron to cytochrome A3, and finally the cytochrome A3 transfer these electron to molecular oxygen. In this day in our country we have seen that there is a shortage of oxygen, and where oxygen is used, it is used in this last step. In this last step the oxygen get reduced and it combined with the hydrogen proton in the matrix and form water molecule. In this way, in the complex 4, when electrons are transferred to the oxygen, 2 hydrogen molecule proton are moved to the intermembrane space. In total we can say that 4 proton from complex 1, 4 from complex 3 and 2 from complex 4, total 10 proton move when the electron from NADH are transported via electron transport chain to the oxygen. So this complete the electron transport chain. So how electron transport chain is connected with the phosphorylation? Before study this, let us take up the example of hydropower project. The chemoosmotic theory is the best theory which describe about the mechanism of phosphorylation. Like in hydropower project, we have reservoir of water. In case of cell, the reservoir of proton is found in between intermembrane space. When when the water flow from in a tube, we have a turbine connected with a generator. When water flow through a channel, it rotates the turbine, and rotation of turbine generates the energy which is used in the household. Likewise, in case of cell, we have ATP synthase. It works exactly like hydropower project. The concentration of hydrogen ion is higher, so there is a tendency of hydrogen ion to flow to diffuse from higher concentration to lower concentration in matrix. What inner membrane is impermeable to these ions, protons, so how they can enter into the matrix. They can enter only through the pore of ATP synthase. When these molecules enter through the pore into the matrix, it causes the rotation of this ATP synthase in clockwise direction. And when this ATP synthase rotates, it brings the conformational change where ADP combined with inorganic phosphate and ATP is synthesized. It is found that to synthesize one ATP, 4 hydrogen ions are required. So we can say that during the transport of electron from NADH to oxygen, we have 10 proton translocated to intermembrane space. So we can say that 2.5, because one ATP is synthesized when 4 hydrogen ions are pumped outside the matrix. So we have 10. So we can say that from NADH to oxygen transport, we can say that one NADH yield 2.5 ATPs. Likewise, when the proton are transferred by a complex 2, in case of FADH2, from there to here, only 4 plus 2 is 6 hydrogen ions are produced, which means it yields 1.5 ATP. So this is the end of our topic. This model explains that how the energy is produced, how the ETC is coupled with the phosphorylation. So this is the end of our topic. We shall meet in next topic. We shall meet in next video with new topics. Till then, take care, stay safe.